And we need cover slips. Do you have a box of cover slips on this table? What happened? Cover slip avalanche. All right, so yeah, thanks. No, I don't want it. You're confusing me. Just keep it there. And do you have the methylene blue over there? Yeah, okay, thanks. Okay, good. All right, ostracods. What is living in there and swimming around are called ostracods. A lot of you kind of looked at them a bit before. They are crustaceans, all right? So you're gonna wanna Google up a picture somewhere, a diagram of the structure of ostracods or the anatomy of ostracods. So when you do actually look at it, you'll have a clue what parts you're actually seeing. Some of you complained to me last week that they were moving around too much. You can do this on your own later. They're moving around too much. Well, don't worry, this week they won't be moving at all. All right? You've got a really good look at parts of ostracods, all right, by the time you finish. So what we have to do here is get some ostracods onto the slide and clean them up a little bit and then immobilize them, all right? So you have at your table, it's kind of mixed up from yesterday's class, but there's some pipettes that are broken off, all right? I did that to get a wider mouth so that it's easier to capture these guys. These are pipettes. They are not fancy pipettes, but they are still pipettes. And they teach you the discipline of not turning over your pipette, all right? You keep your pipette pointing down. If you have some ostracods in there and you do this, where are the ostracods gonna go? Down into the bulb, all right? And you may never see them again. So we try to avoid that, not for the sake of the ostracods, but for the sake of protecting the highly expensive mechanism here. Not really.
20 baht.
No, I think it's more like 20 satang. Okay. But this is just practice for the serious pipettes you guys may have used already or may be going to use, micro pipettes, which really cost $400 up, US dollars up. And if you flip them over, you're out $400 because all your ostracods or materials flow into that mechanism, which is really highly precise, all right? So don't flip these over. Get used to keeping them pointing down. All right, that's one thing. Now, the wide mouth is for capturing them. And at your table, there's also some normal size, narrow ones. Okay, that's for helping to clean them up and maybe adding water or something. Each table has a flask with some plain water at it. If you don't, you can restore your stock of water from the sink. Okay, so let's get some ostracods onto here. Squeeze a little bit. You don't have to do an entire slurp of ostracods. I don't know, what's the right noun for that? A sample that you get by sucking up. A slurp? All right, you don't need an entire slurp. You just need a little bit because these guys are really small. And try to find them in there. Okay. Okay, I see one. Okay, so you kind of carefully, the white paper helps to see them. Okay, there's one in there. I guess I only got one. Now you don't really want the algae this time. There's one in there, can you see it? I'm gonna try to get one more. Maybe they're smarter since yesterday. And yesterday class kind of trained them to be sneaky. I'm not getting very many here. All right, well, I have a few thousand more over there in case. are you? Oh, there's one. Maybe. This is the patience part of it. No. Only algae. Okay, who wants to try? No. You go. Oh, I'll have to do this anyway, so. Oh, I got one, that's it, didn't I? Yep. Mm-hmm. Okay, good. All right, so ostracods are there. Next thing is to clean up the sample. We don't need any algae on there, so just, again, aspirate. The nice word to use in your lab report when you're doing this is not suck it up, it's aspirate. Aspirate some of the liquid away from there. If it's just the culture water, you can put it back into that flask, but you can also have a waste flask on your table. You want to get as much of the algae away as possible. So just kind of aspirating it up into there and then putting it back in the waste or the culture vessel. If you have algae in there, then it will make it harder to see the parts of the animal. Okay. Okay.
Technically, you are killing them, right?
Are they gonna die or?
He's still swimming.
I'm sorry, they will not survive this procedure. They are very simple animals with very uncomplex nervous systems, so I don't think they have a consciousness.
So they don't
right?
feel pain.
I hope not. But in any case, yeah. Okay, so there's your ostracods. I'm going to add a little bit of clean water to spread out the results a little bit. Okay, now your cover slip comes into play, and I will need an eraser if somebody has an eraser. Okay. Now, in this case, you don't have to do that angle thing. You can just drop it on there. And one more. Eee, that one's covered with oil. Why? These used cover slips? You guys never have to reuse cover slips, all right? That one's all right. Okay, drop that one on there. All right, and we take our eraser and we do the squash. Okay? Sorry about that, but that's how it works, all right? That's what you call a squash preparation, all right? So if you feel badly about that, you don't have to do it, all right? You can have me do it, or you can have somebody else do it, or you can just look at the result, all right? But this is part of biology, unfortunately. You have to get a sample somehow.
Are you sure that it didn't get squashed by doing that?
Actually, the point is to squash.
This is something inside.
Aren't we supposed to
just
say
spread
The
around.
squash preparation's job is to take a three-dimensional sample. An ostracod is kind of like a bean shape, and it's too thick to fit in the microscope if you don't make it flat. Okay? So you've got to pancake it, and then the parts will be spread out. And over multiple observations, which involves looking at the diagrams that other people have done for 2 centuries on these things, you can actually figure out what the parts are, which parts are which. You can also do repeatedly and see which parts are associated with which ends of the shell, that kind of thing. But you are basically trying to flatten it so that the light can pass through it and you can see different parts away from each other. Reconstructing it in a 3-dimensional shape is what you do in your head, all right, how they fit together. Okay, so we have a couple ostracods here sampled. I did 2. I think you'll have time to do this. The reason I did 2 is because I want to stain one and leave one unstained. And I think it'd be worth it for you guys to be able
What
to
do you mean
compare them.
stained, unstained?
Yeah, staining is done with something like this, all right? This is a stain, methylene blue. Methylene blue is particularly sensitive to acidic molecules inside the sample, which mostly means actually nucleic acids, all right? So it will adhere to things like mitochondria, which contain DNA, ribosomes, but these can't really resolve ribosomes, all right? but especially the nucleus of cells, all right, will stain pretty well with methylene blue, all right, with a lot of chromosomes. So we're going to stain one of these and leave the other one unstained to compare with. This stuff is concentrated. Another reason you do it over tissue, because it will stain you and stain everything else. Just be careful and just put a drop next to the cover slip, like that. Okay. Wow, you guys are recording everything.
He's recording the video.
Make me so nervous here.
He can record himself.
Yeah. Section 1 is totally different than you guys. They were just like standing around, watching, remembering.
I don't remember everything.
Maybe at the end of the video, you can just
record
take
your
a
own
picture of the
thing.
patient and say,
That's all right.
No, I just record the procedure so I
Okay,
can follow it
so
correctly.
let's get this going here. So your stain needs to reach the animal. Now, some people yesterday tried a different way, which is totally okay. You can put the stain first, or I say put the animal and then put the stain, and then do the squash. All right. I kind of like this way because I think the stain doesn't get loose; it just kind of gets spread out everywhere. But you've got to encourage the stain to kind of penetrate around your specimen there. And I think you guys in the back, can you see how it's going here? It's taking its time to reach into there. You're going to have to leave it for a few minutes. So while you're waiting for that, you actually could be looking at the other one. Just try not to get any stain on the microscope or yourself. It's not really dangerous or anything. Methylene blue is actually used as an antibiotic, a weak antibiotic as a medicine. So a little bit of it is not really going to harm you, but it's just kind of messy. So I'm going to leave that for a while. If you're observing your unstained sample, say for 5-10 minutes, that would probably be long enough for the stain to do its job, okay? Now, let's pretend, okay, cross my fingers, pretend that that's already been 5-10 minutes. What you have to do next is called destaining, all right, destaining. Because after the stain is in there, it's going to stain the cells and so forth. And then you want the stained part to be darker than the background, all right? You want the bluish, and actually it looks kind of purple when you look in the microscope, purplish looking stuff to be on a white background. So that's destaining. In other words, you don't want the extra stain in the water to be there. So the destaining is done with some plain water again. Put it on the same side where you started, and then kind of drive the stain out like that using a little bit of tissue to soak it up. Fold it up, make like a little point like that, and just try to absorb the stain out of there so that and you can do it from the other side as well. You basically want the stain on the biological material, but not in the water around it. That's the idea. And if you don't quite get all of it, it's okay, because you know my specimen is over there, right? It's in the corner.
Stained.
And it seems to be, parts of it are stained already. When you look in the microscope at 400x, how big is the field of view? Can you remember? 450 micrometers. Yeah, 450 micrometers. How big is one of these cover slips in micrometers?
Micrometers? 4,500 micrometers.
This is about 2 centimeters, right? So it's times by
I think it's more like 22 millimeters by 22. A millimeter is 1,000
so 2,000
micrometers.
22,000.
Yeah, 22,000.
So one side of this is 22,000 micrometers. Your field of view is 450 micrometers. Does it matter if you have stain down in the corner here where there's no specimen? Not really, all right? Because you're only going to be looking at a little area over there at the beginning anyway, right? So you can leave some stain on there as long as it's not staining too much. Close that, one of you guys. So let's not too much stain around the specimen itself. All right, so I'm just going to destain a little bit more, try to get that in there. So washing it out. This is also having the effect of spreading out my specimen a little bit. Which is probably a good thing. So the parts are going to be more, not overlapping each other too much. Okay, all right, so that's pretty good. All right, so there you have a stained and an unstained ostracod. Let's see, what I was going to say. Oh yeah, last thing is a tip to make it more observable, you need it to stay wet. Now, all I want you to do for the next 5-10 minutes is get to this point, all right? I'm not going to talk about how to do the oil thing yet. You can, actually, to get to this point, you can actually observe it on there up to, say, 400, all right? You can do up to 400 yourself. You know how to do that already. But when you do start looking at it, you will find out the light is hot enough to make the water evaporate, and it will start drying out, especially from the edges. So if you see a dark line appearing in your field of view, that is not part of the ostracod coming back to life or something. Okay? It is just air kind of coming in from the side. And you should try to add some water, a tiny drop of water somewhere to keep it moist in these kind of wet mount slides, all right? So that's it, all right, that's it really. So that is ready to look at. I think I'm gonna look at it under somebody's microscope right now. You guys go to work, all right? You've got everything at your table, I hope.